Good afternoon, one and all. Myself Swati Pandey from SIRT Pharmacy, Sage University, Bhopal. Today we are discussing a very important topic of lipid metabolism, that is beta oxidation. So lipids, as we know, they are the indispensable for every cell structures, and due to their hydrophobic and non-polar nature, they differ from rest of the body components and they are unique in their nature. The lipids in our body, they constitute about 15 to 20% of the total body structures. They were previously known as the triacylglycerol or triglycerides, which actually constitutes of 80 to 90% of the total lipids. They are also known as neutral fats, and they are stored in the form of adipose tissues in the body. The use of the lipid in our body is to provide an insulating material to the body and regulate the body temperature. It's an interesting thing to know that a 70 kg of an adult, it carries 10 to 11 kgs of fat, that is equal for the production of near about 1 lakh calories of energy. If the same would have been stored in form of glycogen, the weight of a man that would previously be 55 would be increased by the 55 kgs. A normal human being can even survive for 60 to 90 days without food because of the fuel reserve in form of fats in our body. Lipids, they are metabolized by the help of beta oxidation. Beta oxidation is named so because the metabolism takes place at the beta position of the beta carbon of the long chain fatty acids. Coming to the beta oxidation, this is actually a sequential removal of a 2-carbon fragment from acetyl coenzyme A. A 2-carbon molecule, we have a number of lipid molecules. To understand one, we'll take an example of palmitic acid. Palmitic acid is a long chain fatty acid that contains a 16 carbon chain, and sequential removal of 2-acetyl coenzyme A would take place in this. Now coming to the stages of the beta oxidation, the first stage is activation of the fatty acids that takes place in the cytosol. Then this cytosol, where this fatty acid is getting activated, that needs to be transported into the mitochondria. Because the cytosol layer is impermeable to this, a carnitine transport system is being there that helps in transporting the activated fatty acid into the mitochondria. Then the third step, that is the actual beta oxidation, that we call it as beta oxidation proper, that takes place in the mitochondrial matrix. This beta oxidation proper, it again consists of 4 different steps. So fatty acids are oxidized mostly by all the tissues in the body, except the brain erythrocytes and the adrenal medulla, that they cannot utilize the fatty acid for their energy requirement. Coming to the first step of beta oxidation, that is the fatty acid activation. As I said, the fatty acid that we have that needs to be metabolized, it has to be activated. This activation, it takes place with the help of the enzyme acyl coenzyme A. It is activated to acetyl coenzyme by the enzyme thiokinase or acyl coenzyme A synthetase. This is a 2-step reaction, and it requires ATP and coenzyme A along with magnesium ion for this reaction is required. Fatty acid, it reacts with ATP to form acyl adenylate, and then it combines with coenzyme A to form acetyl coenzyme A. Here in this process, 2 high-energy phosphates are released that are further converted into pyrophosphates. This step makes this activation a complete irreversible process, because the 2 high-energy phosphates that were produced, they are immediately converted into pyrophosphates. So this is the enzyme that is required for the activation, that is thiokinase. This thiokinase, it is also consists of 3 different types: short chain, medium chain, and long chain fatty acids. So small chain fatty acids, can be also activated by the enzyme known as thiopurase enzyme. that use succinyl coenzyme A. So this is the first step of beta oxidation, that is activation of the fatty acid to acyl coenzyme A. As we can see, this is the structure of the fatty acid. The number of carbon can vary over here, and acetyl fatty acid. In the presence of the enzyme thiokinase, there is a utilization of ATP which converts into pyrophosphate. This pyrophosphatase enzyme immediately converts it into the two phosphate molecules. This makes the first step irreversible. Again, it cannot be converted back into the fatty acid. So here the AMP gets attached and it forms acyl adenylate. In the second step, there is inclusion of the acetyl coenzyme A, and this acetyl coenzyme A, again, it needs one more ATP, and the product that is formed is acyl coenzyme A. Now, this is our activated fatty acid. Now, this activated fatty acid, it needs to be transported into the mitochondrial matrix. So, for the transportation of the mitochondrial matrix, as I already told you, that it is impermeable to the fatty acids. So, there is a special system known as the carnitine carrier system, or the carnitine shuttle, that operates to transport the fatty acid from the cytosol into the mitochondria. The carnitine, it is actually beta-hydroxy gamma trimethyl aminobutyrate that is synthesized by two enzymes, that is lysine and methionine, in liver and in kidney. So we will discuss the carnitine transport system. This is the carnitine transport system. As we can see, this is the outer portion, that is the cytosol portion, and this is the inner mitochondrial matrix. The inner mitochondrial membrane, here there is the enzyme carnitine along with its carrier protein operates. The acyl coenzyme A, it leaves its coenzyme A, and the carnitine 1 transporter 1, it takes the acyl coenzyme A and it gets converted into acyl carnitine. This acyl carnitine, it gets transported through the inner mitochondrial membrane inside the mitochondrial membrane. Again, in the mitochondrial membrane, this acyl carnitine, it combines with another molecule of coenzyme A, acetyl coenzyme A, to give the carnitine back, and the acyl coenzyme A has now reached into the mitochondrial matrix. So there is a very simple step of transport of this acetyl coenzyme A into the mitochondrial membrane. As we can see, only the carnitine, the carrier protein, now here that has left the acyl coenzyme A, is again transported back into the cytosol. So this is the carnitine transport system. Now our activated fatty acid has reached inside the mitochondrial matrix. Now the proper oxidation will take place. So this beta oxidation proper, it liberates 2 carbon unit of acetyl coenzyme A. So for this, there is a requirement of 4 different reactions, that is OHOC, that is oxidation, hydration, oxidation, and cleavage. Likewise, we can learn it. OHOC, these are the 4 steps that will take place for the complete metabolism of a single fatty acid. So as we are taking an example of palmitic acid, we know that palmitic acid is a 16 carbon compound. So 16 carbon compound will, in the first cycle of the beta oxidation proper, it will remove 2 acetyl coenzyme A. This 2 acetyl coenzyme A. In the second cycle, again it will liberate 2 acetyl coenzyme A. Likewise, for one molecule of palmitic acid to completely metabolize, it will need 7 cycles of this beta oxidation proper to produce 8 acetyl coenzyme A molecules. The first step is oxidation, then is hydration, then again is oxidation, and next is the cleavage. So this is the complete beta oxidation cycle of the fatty acids. Here is our fatty acid, R CH2, CH2. This CH2 that has been marked in red, this is the acetyl, this is the carbon that will be removed in the form of acetyl coenzyme A. This was the first step, that is the activation that we already discussed, that is the conversion into acetyl coenzyme. This acetyl coenzyme, acyl coenzyme, it gets transported from the cytosol inside the mitochondria. Now in the mitochondria, this beta oxidation proper is now taking place. Beta oxidation proper, the first step is oxidation. The oxidation, that is the removal of hydrogen. So in this acetyl coenzyme A, it gets converted into trans-enoyl coenzyme A. Here there is a removal of hydrogen molecule and 2 molecule of ATP is being used here from the the FADH molecule is being used, and this FADH is getting converted into FADH2, that is the generation of energy. This FADH2, it generates energy through the ATP, through the ETC, and each FADH2 is responsible for the production of 2 ATP molecules. Here the enzyme acetyl coenzyme A dehydrogenase is required. So here we can see that there is an introduction of a double bond. Next one is the conversion, that is hydration. Next step of the beta oxidation after oxidation is hydration. So this is the second step, the conversion of trans-enoyl coenzyme A. Again, there is a insertion of a water molecule. So now it has been converted into beta hydroxy acyl coenzyme A, and there is a removal of this one double bond. For this, here we require enoyl coenzyme hydratase enzyme. The third step is again oxidation. This beta hydroxy acyl coenzyme A, in the presence of the enzyme beta hydroxy acyl coenzyme A dehydrogenase, there is a removal of one OH from here and this hydrogens from here, and there is a formation of beta keto acyl coenzyme A. As we can see that there is a removal of one hydrogen from here and one hydrogen from here. This hydrogen removed, it converts NADH into NADH, and this NADH, when it enters the ETC, it produces 3 molecules of ATP. As we have already learned these things in the HMP sheet. So there is a production of beta keto acyl coenzyme A. This beta keto acyl coenzyme A is the last step, that is the conversion of this beta keto acyl coenzyme A, the cleavage into 2 different molecules, that is the liberation of acyl coenzyme A and the acetyl coenzyme A. Here it gets broken and it gets cleaved by the enzyme thiolase. Thiolase enzyme, it cleaves it and it breaks it from over here. So here we can see that there is a production of CH3CO SCOA, that is the acetyl coenzyme A, and the remaining carbon will form acyl coenzyme A. This acyl coenzyme A again will go to the first step, that is again it will follow the whole beta oxidation proper, all the 4 steps, and it will again liberate one acetyl coenzyme A. This acetyl coenzyme A, it enters the TCA cycle. Here the TCA, as we know from each TCA cycle of there is oxidation of the acetyl coenzyme A, and there is a liberation of 32 molecules of 24 molecules of ATP. So if we consider oxidation of palmitoyl coenzyme A, the palmitoyl coenzyme A, it requires 7 coenzyme A plus 7 molecules of FAD and 7 molecules of NAD. As I already told you that for an palmitoyl coenzyme A is a 16 carbon fatty acid, 16 carbon chain. So for that we will need 7 cycles to convert it into 8 molecules of acetyl coenzyme. In the last step, when there will be only 4 carbon after the cleavage step, it will get converted into 2 acetyl coenzyme. So we don't need to go for the 8 cycle. Hope it's clear to you. 8 acetyl coenzyme A and 7 FADH2 and 7. So for the 8 acetyl coenzyme A, that is oxidized by the citric acid cycle, each acetyl coenzyme A will liberate 96 molecules, because for each citric acid cycle of oxidation of one acetyl coenzyme A, it gives 12 ATP. So 12 into 8 is total 96. So the total energy generated from a palmitol coenzyme A is 14 plus 21 plus 96, that is 131 ATP molecules. Now for the utilization, as we saw in the first step, that 2 molecules of ATP were utilized for the activation of the palmitoid coenzyme A. So we'll deduct that 2 ATP. So ultimately, that is the net yield of oxidation of one molecule of palmitate will give 129 ATP molecules. So this is the energetics of the beta oxidation. Now coming to the regulations of the beta oxidation. The beta oxidation, the availability of the free fatty acid, it is the main unit for the net regulation of the net utilization of the FFAs through beta oxidation, as the level of FFA is controlled by the glucagon and insulin ratio. As the glucagon increases, the free fatty acid level increase, and insulin, it has just opposite level on the free fatty acid level. The carnitine transporter 1 is the regulator for the entry of free fatty acid into the mitochondria. Similarly, malonyl coenzyme A, it inhibits the carnitine transporter 1 activity during the de novo synthesis of free fatty acids. The beta oxidation is inhibited. Coming to the first step that of the beta oxidation, that is oxidation. We saw there that SIDS was written. So that is the sudden infant death syndrome. This is an unexpected death of a healthy infant, usually overnight. This is due to the deficiency of medium chain acyl coenzyme dehydrogenase. Glucose is a principal source of energy soon after eating or feeding of the babies. But after a few hours, the glucose level and its utilization decreases. And also, if the rate of the free fatty acid oxidation, that simultaneously must increase so that it can meet the energy needs of the body. Sudden infant death syndrome in infant is due to the blockade of beta oxidation that is caused by the deficiency of the enzyme medium chain acyl coenzyme dehydrogenase. Another disease that is known as the Jamaican vomiting sickness, that is characterized by severe hypoglycemia. Hypoglycemia is the reduced level of blood glucose in the body, along with vomiting, convulsions, coma, and death. It is caused by eating of unripe active fruits that contains an unusual toxic amino acid that is known as hypoglycemia. This inhibits the enzyme acyl coenzyme A dehydrogenase, and the beta oxidation of the fatty acid is blocked, that leads to various complications. Abnormalities in the transport of fatty acids into the mitochondria and its defects in oxidation, it leads to deficient energy production of the oxidation of long chain fatty acids. For example, hypoglycemia, hyperammonia, weakness, and liver diseases. The acyl carnitine, when it accumulates when transferase or the translocase is deficient. Dietary consumption of carnitine, it can improve this conductions. Thank you. I hope the topic is clear to you, and if you all have any doubt, then please comment. We will get back to you to answer your queries. Thank you.